Hello, and welcome to Chapter 22, The Origin of Species. This is the last chapter of the semester, and in some ways we save the best for last, because one thing that we know about evolution is that it is the central theme of biology. We also know that Darwin, the father of modern biology, really was interested in how new species evolved. So let's go ahead and dive into Chapter 22, Part 1, where we will learn about how new species actually form. In order to understand how new species form, we're going to go back a little bit and revisit Darwin's thinking. So remember that Darwin visited the Galapagos Islands, and there he discovered all sorts of plants and animals that were just not found anywhere else on Earth to Darwin's knowledge, and he was correct. They're not found anywhere else on Earth. We could talk about this flightless bird right here, which we mentioned in a previous lecture, but you might ask how a flightless bird got to these isolated islands off of the coast of Ecuador. This is a Galapagos giant tortoise, and the giant tortoise is also unique to the Galapagos Islands. So you might ask how it got there, and that's exactly what Darwin did. And what Darwin's answer was that they got through a process of that simple form of migration that's not gene flow, right, but is genetic drift, and the founding populations. And after new populations were founded, and the speciation events happened. And speciation is defined simply as the process by which one species splits into two or more species. Here's a diagram of how the ancestors to modern whales evolved by becoming, changing over time, becoming more and more and more different species. So when one species splits into two, the two species that result do share many characteristics that they inherited from the common ancestor, but they also differ a little bit from that common ancestor. When we think about speciation, we can kind of compare the concepts that we've been talking about so far of microevolution to this concept of macroevolution. And this is really the first time where we're diving deep into macroevolution. Although I have defined these terms for you before, I'm going to do it again. Microevolution is change in allele frequency in a population over time. Macroevolution refers to the broad patterns of evolutionary change above the species level. So we might think about how groups of organisms such as mammals evolved, or we might think about how the birds evolved, right? That's macro, that's big, large scale evolution that takes place over many, many years. The main trick here for you to understand the difference between microevolution and macroevolution, the only difference that's important is time. Microevolution happens on small scales of time, and macroevolution happens on large scales of time. And we'll talk a little bit more about time and time scales and how we can think about those a little bit later in this lecture. So I've defined species for you on a number of occasions, right? But here we're going to really try to get a strong understanding of the concept of species. So species, you could think about it as the simplest word, and it's like thinking about the Latin derivation of the word species. So species literally means kind or appearance, so type. Biologists put organisms into different species based on lots of different reasonings. For example, the way they look. Now, that word for the way something looks is morphology. It could be physiology or biochemistry. It could be based on DNA sequences. There's all kinds of different ways to organize the species, things into species. But that's just a general idea, a general concept. If we're thinking about the way new species evolve, having this particular definition isn't super easy. I mean, it's not the best kind of definition to be thinking about if we're really focusing here on evolution of new species. And so biologists came up with a very useful definition of species called the biological species concept. And again, this one really applies very well to the concept of evolution. So the biological species concept states that a species is considered to be a group of organisms that live in a population whose members have the potential to interbreed in nature and produce viable, fertile offspring. They do not breed successfully with other groups. So a species is a group of populations whose members have the potential to interbreed. Now, potential, that doesn't mean that every single one of them is going to interbreed with every single other one of them. It just means if they do breed, they can actually breed successfully. That it happens there, that's what discussion of interbreeding. It happens in nature. That means that if we think about, forced confinement or in a zoo or something like that, that's not necessarily just because maybe they can have babies together. It doesn't mean that they're the same species, because it has to be in nature. They have to produce viable and fertile offspring. That viable means healthy, right? And fertile means able to reproduce themselves. And so they're all considered a species if they the same species if they fit into this definition. They're considered different species if they don't fit into this definition. One thing that we can apply right now is this concept of gene flow. Remember that gene flow between populations keeps those populations similar to each other. So these two populations belong to the same species, because with gene flow, their gene pools become more and more similar. Again, it's not saying that there are two different species up here. It's saying they could over time become two different species, but with gene flow, they migrate, they make babies, they stay the same population or the same species. So this here is an example of how we might apply the concept of the biological species concept. Again, it's based on the potential to interbreed. It is not based on physical similarity. So here are two birds that you might look at them and think, oh, they look so similar. They must be the same species. And it turns out that they are not. They do not fit the biological species definition because they don't mate with each other. And the reason they don't mate with each other is because, as you might know, birds sing songs in order to attract mates. And these two birds have very distinct songs, and they just don't find each other's songs attractive. And so they don't mate with each other. So these two birds look very, very similar. Same coloring, same little patches on their neck, same color feathers on their wings and on their chests, but they're different species. On the other hand, we can look at this species and we know that they all belong to the same species, but they're very, very diverse from each other. So, but they have the potential to interbreed and therefore belong to the same species. One of the ways that you get new species is through reproductive isolation. This is so important because remember that as part of our definition of the biological species concept, we say that you have to be able to have the potential to interbreed. So reproductive isolation is the existence of barriers that stop two species from producing viable, fertile offspring. So that's anything that stops that reproduction, and the production of viable, fertile offspring is called reproductive isolation. And we can also introduce this term hybrids, because we'll be talking about this throughout this lecture too. Hybrids form when the barriers that limit production, those reproductive isolating barriers, that they are broken down. And so you form hybrids between two species. We'll talk about the importance of hybrids later. So reproductive barriers can be classified by whether or not they act before fertilization or after fertilization. Let's take a look at some of these barriers that happen before isolation. So we call any barrier that happens before reproduction, we call that a prezygotic barrier. Pre meaning before, and zygote or zygotic, but zygote, right? You've heard that word. That's when egg meets sperm and you have a fertilized egg. So a prezygotic barrier stops fertilization from occurring. And there's several different ways that this can happen. It can happen by preventing mating itself. It can stop even attempting from mating. It can prevent the successful completion of mating, or it can stop the fertilization process, even if mating does happen. So we'll go through a bunch of different examples and give you some new terms associated with prezygotic barriers. In the first one, again, we're talking about reproductive isolation, right? The first one is called habitat isolation. With habitat isolation, two species, they just don't really encounter each other. If they do, it's very rarely, and that's because they are inhabiting different habitats. Even though they live in the same general location, their habitats are different. So there's no physical barrier keeping these two flies from encountering each other. It's just that this one prefers one habitat and this one prefers another habitat. If we get more specific, these are fruit flies, and fruit flies lay their eggs on very specific fruits. This one right here lays its eggs on apples, and this one here lays its eggs on hawthorn fruit. Now you can find apple trees and hawthorn trees, within a fairly short distance of each other, but these flies just kind of hang around the apple trees, and these flies just kind of hang around the hawthorn trees. So they have habitat isolation. The next example of reproductive isolation is temporal isolation, and temporal refers to not a delicious meal you can get at a Japanese restaurant, but time. So temporal isolation refers to species that breed at different times of the day or different seasons or in different years. You can think about like cicadas. Some cicadas emerge every 13 years and some emerge every 17 years. Those cicadas are isolated by time, so they have temporal isolation. And a good example here is seen in these two species of skunks. Yes, these are different species of skunks. You might not be familiar with them, but they are, in fact, skunks. So these two skunks do overlap in geographic regions, just like the fruit flies that I showed you on the previous slide. However, one of them mates in late summer, that's this one right here, and the other one mates in late winter, that's this one here. So if this one's only mating in summer and this one's only mating in winter, they're isolated by time because they aren't able to mate with each other because they mate at different times of the year. A third kind of reproductive isolation mechanism is called behavioral isolation, and this is seen in different courtship rituals. We talked about this with the yellow birds from the previous slide, right? They have different songs, and so they don't mate with each other because they're just not kind of interested in each other. A really cool example of this can be seen in this bird right here, the species called the blue-footed booby. And blue-footed boobies have very, very specific courtship rituals. I believe your your book, your ebook comes with a really nice video that will show you this courtship ritual if you are interested. But one of the things they do is they lift up their feet and they show each other the bottoms of their feet, and they do these head bobbing dance, and it's very, very interesting. But of course the female is really only going to mate with a male that does this very specific dance. That video that is on, that comes with your book, again, definitely worth watching. I have a short version here for you of giraffe courtship, and I like this one just because it's it does show just a brief video, and I don't like to show you videos within videos. So here we go, the giraffe courtship.
Plain in Africa are bumping each other's necks in a courtship ritual. Courtship usually involves a complex set of behaviors believed to serve as confirmation that the participants are of the same species but of opposite sex, are in good physiological condition, and do not threaten each other. Credit National Geographic.
So a nice short video just showing you giraffes' courtship rituals. I also wanted to bring your attention to this other video showing prezygotic barriers to mating in Galapagos finches, and I invite you to watch that in your textbook too to help you understand these concepts of prezygotic barriers. So the ones that I've mentioned so far, these reproductive isolating mechanisms, they actually stopped an attempt for mating, right? You have to have the right behavior, or you have to have mated at the right time, or you have to be in the right habitat. But what if you don't meet, you're two species, but you don't meet any of those, and you try to mate? What happens next? Well, you could have mechanical isolation. Mechanical isolation happens when, and I'm going to be graphic here, it's when the bits and pieces, the reproductive organs of one species does not fit into the reproductive organs of another species. And a very interesting example that you can see here is in these two different species of snail. One is a right-handed snail and one is a left-handed snail. And you can tell right-handed snails from left-handed snails looking at the curve of their shell. So this one curves to the left and this one curves to the right. So when snails mate, they approach each other head on, just like you see right here. They're approaching each other head on. And when they do this, they move their heads past each other, like you see here. They move their heads slightly past each other. And when they do this, their genitals emerge and they basically copulate, right? And this is snail sex right here. But if you have a left-handed snail and a right-handed snail coming at each other, their genitals are basically, they're on opposite sides of their body, which as they're coming at each other head on, it looks like they're kind of on the same side. And so they can't actually copulate, right? They physically can't copulate because their genitals just don't reach each other, right? Remember that snails copulate head on like this. So their genital openings don't align and they cannot meet. That's mechanical. That's an example of mechanical isolation. There's like something physical about them that does not allow mating. The last of the prezygotic barriers is called gametic isolation. Now, these are prezygotic reproductive isolating mechanisms, right? So the example that we're going to step through here is found in sea urchins. And this is sea urchin. It kind of looks like a flower, I know, but it's a sea urchin. Sea urchins have these very sharp and long spines coming off. You definitely would not want to step on this. And it's a marine animal. So with gametic isolation, we see this in a lot of marine animals. What happens with sea urchins is that the males and females, they kind of just release their sperm and eggs into the water. And if they do this at the same exact time or in the same location, the sperm is going to meet up the egg and they're going to have fertilization. But this is two different species of urchins. This is the red urchin and this is the purple urchin. they both release sperm and egg into the water around the same time. And so the sperm and the egg try to fuse together, but they can't do it. And this is because the proteins that are found on the outside of the surface of the eggs and the proteins that are found on the outside surface of the sperm, they don't allow the fusion to happen. So the proteins don't bind together very well, and so fertilization just doesn't happen very often. So this is gametic isolation. The gametes themselves are what's keeping those two species as separate species. The next thing that we're going to talk about is postzygotic barriers. So far, we talked about the 5 different kinds of prezygotic barriers, and now we're going to talk about 3 different kinds of postzygotic barriers, and those are called reduced hybrid viability, reduced hybrid fertility, and hybrid breakdown. Postzygotic barriers exist when a hybrid, an offspring, is formed because an egg and a sperm fuse together. So they fuse, but the hybrid either doesn't result in a full-grown adult that's also fertile forming, or something else happens. And I'm going to step you through each of these 3 right now. So let's start out with reduced hybrid viability. Again, this is a postzygotic isolating mechanism. Reduced hybrid viability is seen in the salamander right here. This is a salamander. You can see those huge salamander eyes and cute little salamander feet. What happens in different species of salamanders is that the genes of the different species, they kind of interact in these very weird ways that reduce the ability of the offspring to develop properly. So in this salamander right here, it lives in the same region and same habitats with other salamanders. Occasionally they do hybridize, but those hybrids do not finish the developmental process. They kind of either die in the egg, or they're just very, very frail newborn babies, and so they die at that point. Reduced hybrid viability. The next one is reduced hybrid fertility. So what you see with reduced hybrid fertility is that you might be able to have a hybrid between 2 different species, and those hybrids are healthy and vigorous, but they might be sterile. And if they're sterile, they're exhibiting reduced hybrid fertility. So, for example, here you have a donkey, and this is a horse. This is actually a male donkey, and this is a female horse. This becomes important in just a second. And when a male donkey and a female horse mate, they have a mule as the offspring. Now, mules are very healthy, they're very strong, but they cannot themselves reproduce. They are infertile. You also might say sterile. If it was the other way around, if it was a female donkey and a male horse, then you would have something called a hinny. Not a mule, but a hinny, and hinnies are also sterile. So that's an example of reduced hybrid fertility. The last example is called hybrid breakdown. And with hybrid breakdown, it actually takes more than 1 generation for the hybrids to start showing bad signs. So the first generation hybrids are fertile and they're vigorous, but when those first generation hybrids mate with each other, then you get the second generation. Or maybe they're, the first generation hybridizes with the parental species. Then what happens in the next generation, the second generation, you could think about that as the F2 generation maybe, is that they're feeble or they're sterile. So this is rice. I don't know if you've ever seen rice before on a plant, but this is rice. And we have 2 different strains of rice, and they are different strains, and they're different strains because they've accumulated, different mutations. This is very common. Each of these different strains has accumulated a different set of mutations, and that's after they diverged from a common ancestor. So, yes, you can have hybrids between them, and they're vigorous and they're fertile, but in the second generation after that cross, they have too many of the recessive alleles, and so they're small and they're sterile. We don't officially call these two species separate species. We just say they're different strains, but we also do say that they're in the process of evolving into different species. And the reason that they're in the process is because although the F1s are fine, the F2s are not fine. Well, I hope this makes sense to you about how species is defined and how species are maintained as separate through reproductive isolating mechanisms. When we come back, we're going to get a few other definitions of species and look at the role of geography in the formation of new species.